So in this particular study, we are looking at evolving genomic landscape in metastatic prostate cancer patients, especially patients who have received 3 different types of treatments, including taxanes, ARPs, and PARP inhibitors. And we matched the samples and made sure to only include those patients who had a next-generation sequencing through Guardant, as the study was through Guardant-informed database as well, exactly before starting the treatment, and on progression within 90 days, a second NGS sample, and comparing both the samples. So overall, looking at the entire study, what we saw was there are many new mutations, and particularly pathogenic mutations, which are arising after patients have progressed on these treatments. So that was like an overall aim to look at how this genomic landscape is evolving for all these patients. In the poster that I'm presenting at ASCO GU, we particularly focused on HRR alterations, and for sure we would think that there would be some link between HRR alterations with PARP inhibitors, but we also saw that there are new alterations arising in BRCA1, BRCA2, and other HRR genes as well, after taxanes as well as after ARPs. In PARP inhibitors particularly, we saw some of the overall prevalence, maybe if it reduced for BRCA2. However, there were many new alterations which arose after the treatment or when the patients progressed. So all these new mutations kind of make us think, what next? And then it also makes us think, are these alterations kind of specifying that it is prognostic, or it's just that evolving biology of the cancer?